Aside from carbohydrates, lipids are also a key form of storage reserve in the seed. In this lecture, we will look at the process by which lipids or stored fats in seeds are broken down to provide the embryo with the nutrition it needs during germination. Lipids or fats are stored in the seed in the form of triacylglycerols or tags. Triacylglycerols are made up of a glycerol molecule attached to three fatty acid chains. This figure illustrates the basic structure of a triacylglycerol. We have here the glycerol backbone, and attached to it are three fatty acid chains, 1, 2, and 3. These fatty acid chains can either be saturated or unsaturated or a combination of both. So what do we mean by saturated or unsaturated fatty acids? We have here on the right examples of saturated and unsaturated fatty acids. On the top, we have here a linear carbon chain that is attached to the highest number of hydrogen atoms possible. In other words, the carbon chain is saturated with hydrogen. A carbon chain that is saturated with hydrogen atoms is a saturated fatty acid. Now below, we can see a carbon chain having two carbons attached to each other with double bonds, here and here. Because these two carbons have double bonds and are not saturated with hydrogen bonds, we describe this fatty acid chain as unsaturated. During seed germination, triacylglycerols need to be broken down into its component glycerol and fatty acids before it can be metabolized. Within the cells of storage tissues, triacylglycerols can be converted to sucrose for transport to other tissues in the growing embryo. Within the seed, the triacylglycerols are stored in the oil body or oleosomes. As I mentioned earlier, the triacylglycerols need to be broken down into its component glycerol and fatty acids before it can be fully broken down and used as energy by the embryo during germination. The first thing that needs to happen is for the enzyme called lipase to hydrolyze or break down the bond that connects the glucose from the three fatty acid chains. The lipase enzyme is produced in the protein envelope of the oil bodies or oleosome. After cutting or breaking down the triacylglycerol molecule to its component glycerol and fatty acids, the glycerol molecule will then be transported to the cytosol, where it will be converted to sucrose through the process of glycolysis. What will happen to the fatty acids that were cleaved from the glycerol molecule? The fatty acids will go to the peroxisome. Peroxisomes are membrane-bound organelles in the cytoplasm of eukaryotic cells that contain oxidative enzymes such as catalase and peroxidase. Once the fatty acids are in the peroxisomes, they will further be degraded via two pathways, alpha oxidation or beta oxidation. We will be discussing these two pathways in more detail in the succeeding slides. Now, the mechanism of transfer of fatty acids from the oleosomes to the peroxisomes is unknown, although peroxisomes are usually found adjacent to oil bodies. In this panel, the oleosomes or oil bodies appear as light staining structures that are located adjacent to the peroxisomes. The oil bodies are labeled as OB and the peroxisomes are labeled as P. This physical closeness of the oleosomes to the peroxisomes within a cell could facilitate transmembrane transport of the fatty acids from the former to the latter. Once the fatty acids are in the peroxisome, they will be broken down into simple sugars through the process of alpha or beta oxidation. Alpha oxidation pathway is called as such because the first or the alpha carbon of the fatty acid chain is the carbon that undergoes oxidation first. The fatty acid chain is further broken down until it is reduced to a molecule with only 12 carbon atoms. The successive loss of one carbon atom and carbon dioxide is facilitated by the peroxidase and aldehyde dehydrogenase enzymes. Alpha oxidation is not the main pathway for triacylglycerol breakdown in seeds, so for the purpose of our discussion on triacylglycerol metabolism, we will focus more on the beta oxidation pathway for tag breakdown. How is beta oxidation different from alpha oxidation? Beta oxidation is a cyclic process that serves as the major means of fatty acid breakdown in seeds during germination. Under this pathway, the fatty acids are broken down to the two-carbon molecule acetyl-CoA. The process is called beta oxidation because the number 2 or beta carbon of the fatty acid chain is the carbon that undergoes oxidation first. Let's look more closely at what happens during degradation of triacylglycerols via the beta oxidation pathway. Let's start with the intact triacylglycerol. Even before the seed enters the second phase of water uptake, enzymes that are necessary to break down storage reserves, including lipids, are going to be activated. These enzymes include the lipase enzyme. The job of the lipase enzyme is to break down the triacylglycerol, which is the stored lipid form, into its glycerol and three fatty acid components. The glycerol molecule will go to the cytosol, where it will be broken down to simple sugars by the process of glycolysis. The fatty acids will then move to the peroxisome, where it will undergo degradation via beta oxidation. This is the basic structure of a fatty acid. We have here a carboxyl or functional group and a hydrocarbon chain or R group. If we expand our R group, this is how it's going to look like. It will be composed of a carbon chain attached to hydrogen atoms. This R group is variable depending on the number of carbon chains. In this example, we have an 18 carbon chain which is characteristic of a stearic acid. If the carbon chain is composed of only 16 carbons, that is palmitic acid. If it only has 14 carbons, then we have myristic acid. Aside from the number of carbon atoms, the fatty acid chains will also vary based on the presence or absence of double bonds. Let's go back to the process of beta oxidation as a means to break down stored lipids. I said earlier that the process is called beta oxidation because the number 2 or beta carbon of the fatty acid chain is the carbon that undergoes oxidation first. This is just an enlarged version of the fatty acid that we examined in the previous slide. This fatty acid has 18 carbons without double bonds, so this is stearic acid. We have here the carboxyl group, and then over here, the hydrocarbon chain or the R group. The first carbon following the carbon of the carboxyl group is the alpha carbon. The second carbon is called the beta carbon. When this fatty acid undergoes oxidation, this beta carbon will be the first carbon to undergo oxidation, hence the name beta oxidation. Let's now see how these fatty acids are actually degraded. Let's use stearic acid as an example again. The stearic acid that is released from the glycerol molecule and transported to the peroxisome is still inactive, meaning it cannot be degraded by enzymes. Before it can be accessible to enzymes, it needs to be activated first. This is possible by replacing the OH group in the carboxyl end with a coenzyme A. This is facilitated by the enzyme fatty acid-CoA synthase. Once we have the coenzyme A or CoA, our hydrocarbon chain is no longer stearic acid, but stearoyl acyl-CoA. This activated fatty acid can now undergo beta oxidation. Activated fatty acids are oxidized in a four-step cyclic process within the beta oxidation pathway. The final products of beta oxidation are two carbon molecules in the form of acetyl-CoA. Every pass of a fatty acid through beta oxidation yields one acetyl-CoA and a fatty acid chain that is two carbon molecules shorter than the original fatty acid that entered beta oxidation. So say for example, we start with stearoyl acyl-CoA, which is an 18 carbon fatty acid chain. After passing one cycle of beta oxidation, our products will be 1-acetyl-CoA, which is a 2-carbon molecule, and palmitoyl-CoA, which is a 16-carbon chain. Palmitoyl-CoA is 2 carbon molecules shorter than stearoyl-CoA. The resulting palmitoyl-CoA still needs to be broken down to smaller molecules. Remember that the final products of the full beta oxidation of a single fatty acid are 2 carbon molecules in the form of acetyl-CoA. So if the palmitoyl-CoA product of the first beta oxidation goes through another round of oxidation, This will again yield one acetyl-CoA and a carbon chain that is two carbon molecules shorter than what we started with. In this case, from the 16 carbon chain, we now have a 14 carbon chain resulting from beta oxidation. This 14 carbon chain is called myristoyl-CoA. Let's look at the enzymatic reactions involved in the actual beta oxidation process. There are four reactions involved in beta oxidation, and these are oxidation, hydration, dehydrogenation, and oxidation reactions. The first oxidation reaction is catalyzed by the enzyme acyl-CoA oxidase. In this example, we have a 16-carbon fatty acid that has already been activated. We have the carbon shaded as an indicator of their position relative to the carboxyl group. The carbon shaded in pink is the alpha or first carbon from the carboxyl group. The carbon shaded in blue is the beta or second carbon from the carboxyl group. During the oxidation reaction, there is an addition of oxygen atom that leads to the removal of hydrogen atoms from the beta and the alpha carbons. Consequently, this generates heat as well as hydrogen peroxide, which is harmful to the cell. To prevent the accumulation of hydrogen peroxide, another enzyme, known as catalase, immediately cleaves hydrogen peroxide to produce water and oxygen. The first oxidation reaction results in a beta carbon attached to the alpha carbon with a double bond. This product is called trans-enoyl-CoA because the two hydrogen atoms are now in a trans state with each other. The second step in the beta oxidation process is the hydration reaction. This is facilitated by another enzyme known as enoyl-CoA hydratase. In this step, a water molecule is added between the beta and the alpha carbon. With the addition of water, a hydroxyl group is now attached to the beta carbon, and the alpha carbon is again saturated with hydrogen atoms. This product is known as hydroxyacyl-CoA. This hydroxyacyl-CoA will then undergo a dehydrogenation reaction that is catalyzed by the enzyme beta-hydroxyacyl-CoA dehydrogenase. During this step, hydrogen is removed from the beta carbon, producing a double bond between the carbon and oxygen atom. This reaction converts the hydroxyl group into a keto group. The resulting ketoacyl-CoA is a more symmetric and balanced molecule that can be cleaved to produce two molecules. Cleavage of this molecule requires coenzyme A and is catalyzed by the enzyme acyl-CoA acetyltransferase, or thiolase for short. This final reaction is also called thiolysis in reference to the shortened name of the enzyme that catalyzes the reaction. With the activity of thiolase, the ketoacyl-CoA is broken down to one 2-carbon molecule known as acetyl-CoA and a 2-molecule shorter version of the original activated fatty acid. The resulting 14-hydrocarbon chain will then re-enter the oxidation cycle to be further broken down. One pass through the 4-step cyclic process of oxidation removes one acetyl residue in the form of acetyl-CoA. So say for example you have a 16-carbon fatty acid. It needs to pass through 7 cycles of beta oxidation to get fully oxidized and produce 8 molecules of acetyl-CoA. In summary, stored lipids in the form of triacylglycerols are broken down by lipases into their glycerol and fatty acid chain components. The glycerol molecule is transported to the cytosol, where it will be broken down into simple sugars through the process of hydrolysis. The fatty acids, on the other hand, are transported to the peroxisomes, where they will be broken down through alpha or beta oxidation. Alpha oxidation breaks down fatty acid chains to a 12-carbon molecule. Beta oxidation breaks down fatty acid chains to 2-carbon molecules known as acetyl-CoA. In germinating seeds, the breakdown of fatty acid chains are mostly through beta oxidation pathway. Beta oxidation involves four enzymatic reactions, namely oxidation, hydration, dehydrogenation, and oxidation.